All right, so the first question we're going to deal with, we just dealt with on the quiz. How does each RNA type function in translation? I think it's a good reminder to reflect on that in transcription, you're dealing with every single type of RNA has to be transcribed. Many of those are not going to be involved directly in making protein product. And then in translation, you have 3 different RNAs involved in making some kind of a protein product. So DNA to RNA transitions, that transcription event is fairly simple. The language doesn't really change. The nucleotides you're choosing is different. They have different sugars and a different base in the case of thymine and uracil. But overall, the language really isn't changing. Translation is more complex. And so to go from RNA to proteins, it's fairly complex, happens outside of the nucleus in eukaryotic cells. And so it involves a great deal of work. First, within the mRNA, so our first type of RNA involved in translation, mRNA. Within the mRNA, you have what's called the genetic code, a series of 3 bases that represent a specific amino acids. So every 3 nucleotides, we call this a codon, a language where one word has 3 letters and codes for a single amino acid, where you have a word with a single letter. And so this role or this part of translation is taken care of by mRNA, okay, bringing that code. mRNA is the RNA complement of your template strand. Remember when we talked about transcription, we talked about in many ways it's similar to copying the DNA, to making copies, except for you are writing an RNA complement rather than a DNA complement. And you're also not writing the entire length of the strand, but instead just a short portion of it. Then tRNA is the RNA that brings the amino acids. So T is sometimes referred to as translation RNA or transfer RNA. It is bringing the amino acids and transferring them to the growing polypeptide chain. It is what's actually providing the translation. And it does this because on one end of the tRNA, there's what's called an anticodon. 3 bases that are complementary to the codon. So codon is a string of 3 nucleotides that represents a word. The anticodon, a string of 3 nucleotides that is complementary to the codon, and translate that that word that's made up of 3 letters into a word made up of a single letter, your amino acid. And then finally, ribosomal RNA, rRNA, makes up ribosomes and is actually catalytic. It provides the catalysis for the reaction of attaching a new amino acid to the growing polypeptide chain. And this is interesting because up until researchers discovered that it was RNA doing that catalysis, nobody knew that RNA could actually catalyze reactions and thought it was only proteins that could catalyze reactions. And this is neat because now you have RNA can store information and it can actually catalyze reactions, lowering activation energy. So RNA can do the function of both DNA and of proteins in a single molecule. It's exciting stuff. It's exciting stuff. And so, yeah, you've got 3 different RNAs integrally involved and intimately involved in this process of translation. And so here's kind of an overview of translation. You've got your ribosomal RNA making up these ribosomes. You've got your tRNA or transfer RNA bringing your amino acids in. And you've got your mRNA providing what gets translated, providing the codons. All right, so we're going to do a lecture break right now. And I know we've done a quiz, so we haven't been talking all that much. But I would like for you to spend a couple of minutes talking with your group about why it's exciting to have a molecule that can both store information and catalyze reactions. Why is that exciting, that RNA can store information and catalyze reactions? That is, do the work of both DNA and proteins. Okay, why is that exciting? Take a couple of minutes talking about it with the people in your group, and then we'll talk about it as a bigger group.
And they can function
store information and catalyze. Yeah, store information, catalyze reactions.
Right. Well, from it.
Yeah, basically.
It's okay. And then, like, they say the original. Subjective.
So tRNA, does it actually store information? Because it seems to be.
Sure. Yeah. We'll talk about that in a minute. All right, you got about 10 seconds to wrap up your thoughts and get somebody to represent your group to the class as a whole. All right. So we're going to start here in group one, and we're going to work our way clockwise. So group one, do you have some ideas on why that's exciting? What's exciting about having a molecule that can both store information and catalyze reactions?
We said that it adds another level of complexity.
Sure. Yeah, and that it doesn't, all of the information storage doesn't have to be in the form of DNA, and all of the enzymatic activity, catalyzing reactions, doesn't have to be done by proteins. Good, I love it. Group two, you lost a member. 2 members. Well, just one today, right? Or did you have 2 that both left today? Yeah. Do you have some thoughts? We'll take a pass. Okay, group 3.
We said that it was like, I feel like it would better facilitate structures within the cell to have RNA that can kickstart protein function.
Sure.
It goes from DNA to RNA, from RNA to protein.
Mm-hmm.
Have something that can also carry genetic information as well as facilitate various reactions within the cell. Like that is more beneficial for the cell as a whole instead of only like I don't know. Because proteins also facilitate reactions, but I feel like having multiple people doing one
Sure.
job can help.
Yeah, it creates what you would call redundancy, right? To where the actions can be carried out by multiple pieces. And so if one fails, you still have that other partner that can kick in. Yeah, it's called redundancy, and you see it a lot inside of cell function. Group 4. Is there conflict between groups?
No.
Oh, okay.
I think we kind of talked a little bit about it's more efficient
Okay, how so?
for the cell because if you didn't have one that could also catalyze the process, then you'd have to have an additional protein.
Right, so
to catalyze.
yeah, so what's actually doing the catalytic work in translation is a ribosome. Or I'm sorry, is a ribosome, but is an RNA that doesn't have to be translated by a ribosome to serve that function, right? So it can carry out that enzymatic function without having to be translated into a protein by a ribosome. And so that's nice, right? What's actually doing the work of translating and forming proteins isn't necessarily a protein. So you don't have like a which came first, the chicken or the egg problem there, because what actually is translating protein is not a protein. although the ribosomes are made up of both proteins and RNA and can't be made up of just RNA. But what's actually doing that work isn't a protein. Group 5.
We also said efficiency.
Okay, efficiency. Group 6.
Pretty much the same thing, just less steps in the process.
Okay.
Okay, so the other thing we said though was like you can have a much simpler organism that
Sure.
only has RNA.
Right. It gives you the potential to have an organism that doesn't have DNA and doesn't have proteins. That it's storing its information as RNA, and the RNA is catalyzing all of the reactions necessary for that organism to live. And you have that all being done in a single molecule. And this is where it's really interesting. And so you have an idea right now with regards to the origin of life conundrum, is that maybe your original survival machines had only RNA. And you have what's called an RNA world, where you have survival machines, you have organisms they store their information as RNA, and they use RNA to catalyze all the chemical reactions they need to survive. And then over time, the reason why you develop DNA and proteins is because the organisms that begin to develop that have a fitness advantage over organisms that don't. But originally, you don't. The reason why this was exciting is because you don't have to have DNA and proteins in order to think about an organism having the characteristics of life. You could have a simpler design and still have something that would be characterized as a living organism. Okay? It's exciting. There are some problems. One, we don't find any RNAs that both store information and are enzymatic. They're two different RNAs, right? Our RNA is enzymatic, tRNA, mRNA store information. So that's one problem, right? You still need two different types of RNA. It's not quite as simple as like, hey, the same molecule can do both. Yeah.
What about in viruses? They have like different RNA.
Yeah, but viruses aren't living organisms, and they can't self-replicate, they can't metabolize, they can't respond to stimuli, they have to manipulate host machinery to do all of that. And so when you talk about the characteristics of life, having DNA is one of the characteristics of life. As far as we know, there are no living organisms that store their information as RNA. Right? Viruses do, but they are not living organisms. Because here's another big problem with this idea. RNA is very unstable. RNA is very unstable. DNA, incredibly stable. Right? You could take DNA, you can bury it under a rock and come and find it 100 years later, and you're going to be able to have almost 100% fidelity. You're going to be able to get, just if you were to go and sequence it, you're going to have a pretty good probability of knowing exactly what the sequence was after 100 years. RNA, you leave it in a test tube for 30 minutes, and it deteriorates like crazy. It's very unstable. So that's another big problem. And you're like, well, that would make it to where organisms that start to make DNA are now favored over organisms that store their information as RNA because RNA is unstable. Yeah, but RNA is not even stable enough to really store information from one generation to the next. It's just not. And that's why living organisms don't store information as RNA from one generation to the next. I mean, viruses will do, but they'll assemble, and if viruses don't find a host right away, the virus dies. Right? Which is why if you cough on a wall and you leave it there for a couple of days and somebody goes and licks the wall, they're not going to get sick. Don't do that. Don't try it. It's disgusting. Plus, somebody will probably come and lick the wall before enough time has passed. Yeah.
Okay, so is it even probable that the original survival machines had used RNA?
I would suggest that it would be better to suggest that they had something completely different than what we have now. They didn't use RNA, they didn't use DNA, they didn't use proteins. They used something completely different that just got completely replaced by what we have now. Because RNA is too unstable. I mean, it's just too unstable. You can have something that has more stability and is still self-replicating like nucleic acids are. But yeah, I think would be better to go with something completely different. Plus, if you're going to form DNA from RNA, what do you think you need? You need something to read it, and what's that thing going to be likely? It's going to be a protein. And if you're going to go from RNA to protein, and you're like, well, it's cool, ribosomal RNA is enzymatic, so you don't have to go to protein, but if you want to go from RNA to protein, what do you need? A bunch of proteins. a bunch of proteins to make that happen. So then we also have missing mechanisms. Was there a hand back here, Charles?
Yeah.
Were you going to ask how is tRNA information storage again?
No.
Okay. Yeah.
Oh yeah. Why do organisms use RNA in the first place instead of everything being DNA?
Well, with eukaryotic organisms, you have to, because DNA is too big and bulky to leave the nucleus to shuttle in and out. Like our chromosomes are way too massive to move them out to have it be translated. And then even if you were going to say, okay, well, let's just copy a small portion of it, but with a DNA complement instead of an RNA complement, and we'll just ship that out. The problem is it's too stable. You don't want something sticking around that long because it gives the cell less control over how much expression happens. If it ships out something that's really stable and is going to stick around for a long time, it's going to have to invest a lot more energy into controlling that expression than if it ships something out that's unstable and is only going to express for a certain amount of time and has a short half-life, right? Especially since conditions change all the time. Cells are in dynamic environments where conditions are always changing. It's like it needs something at 1 minute and doesn't the next. Where in reality, it needs something at the 1 nanosecond and doesn't need it the next, right? And so the conditions are always changing. Yeah, Sadie.
Is it also, with the not using the DNA, is that also better for like, for the cell to not, like, isn't that easier to help prevent, something going wrong, like some?
Yeah, but if you're going to make a copy, so you would still have your main molecule, and you're just making a copy of it. So you're working with a copy and not the original, right? So you could still do that while making your copy DNA rather than RNA.
So it's like the photocopy where it's like you have your original and then you're
Right, your original's safe and protected within the nucleus, and you're working with the copy. I mean, it's a good copy. It's a great copy. And so you could do anything with the copy that you could with the original, but it's not the original. And so it's not as devastating if there was a copying error that was made in the process. Yeah. So you could accomplish that even if you were transcribing into DNA and not RNA. The main reason is the stability. Yeah.
Is that why when we talk about DNA replication, there's a lot of processes to fix errors?
Yes.
Whereas we don't see those in proteins.
Right, you're right. Yeah, and so when proteins are made, there really aren't mechanisms to fix errors. If you just have proteins that are made in error, they just get recycled. It's just like non-functional protein. Tag it for destruction. There aren't as many mechanisms to ensure fidelity. You're right about that.
Okay.
Yeah. And that's because it's not as essential. It's not storing information. So tRNAs are RNAs that actually form hydrogen bonds. So you get one long RNA transcript, usually somewhere between 75 and 80 bases long, and then it folds back on itself and forms a three-dimensional structure. And then one side of that is the anticodon, and the other side needs to be charged. It needs to have an amino acid added to it. And Charles, this is the answer to your question. How do tRNAs function to store information? Within their sequence, they designate to these amino acyl tRNA synthetases, these are enzymes, what amino acid to attach to it. Because they have within their sequence the information to tell this protein which amino acid to attach to it, which amino acid to charge it with. And so they do store information in that regard. Now, ribosomes are constructed of both ribosomal RNA, 4 different types of ribosomal RNA, and a number of different proteins. And so they have both a small subunit and a large subunit. Which one binds first in translation? Small or large? Small. Small binds first. Small binds the mRNA first, and then the large gets recruited later on. That's an important piece to keep in mind. And not for this Friday, because Chapter 15 is not on this midterm, but on the next midterm, which will come sooner than you know. Y'all feel like the semester's going by pretty fast so far? I am. Man, it's just full speed. Full speed. All right? So tRNAs and rRNAs, both vital to the function of translation, and both really have a very specific structure that allows them to carry out their function. And so here is a tRNA. Here's the two-dimensional structure. And again, it's usually between 75 and 80 total bases that then folds back on itself. And so at one end, you've got your anticodon with a series of 3 nucleotides that are complementary to the 3 nucleotides in a codon, but will only match really well with 1 codon. And then at the other end, you charge it by attaching an amino acid. And that's done by an aminoacyl tRNA synthetase, which is a protein that charges tRNAs. So that after the tRNA transfers its amino acid, it can go and get recharged and bring back another one, and it can go and get recharged. But because RNA is unstable, it's not going to last forever, and so the cell is constantly making more tRNA using RNA polymerase 3. RNA polymerase 1 involved in your ribosomal RNA. Yeah.
Why does it need so many bases?
No, that's
or
not
that
true.
Specific shape?
Oh, sorry, go ahead.
Why does it need that many bases or that specific shape?
Why does it need so many bases? It has to be big enough. Yeah, it's a good question. It has to be big enough that it can interact with the protein, the aminoacyl tRNA synthetase. So it has to be big enough that it can be recognized by that protein, and that protein will bind it to attach an amino acid on it. If it was really small, it's going to be really hard for it to bind with any particular protein and to actually have separate domains. And so being large enough to actually where it can have 2 separate domains. Yeah.
By charge, do you mean just provide energy? Or like,
By charged, it means it has that amino acid attached to it. It's not charged like it's given 2 extra phosphate groups to provide energy. Although we use that same language for nucleotides as well, because nucleotides with just a single phosphate group, if they're going to then be used in transcription or copying, have to be charged by having 2 additional phosphate groups added to it.
So charge just means added to.
Yes. Yeah. Yes. Okay. And so here's kind of more of a 3-dimensional structure to tRNA with its amino acid attached to it. And here's the process of aminoacyl tRNA synthetase adding an amino acid to this tRNA. And notice in the process it requires energy to do that, but it's not charging the tRNA with energy. It's just charging the tRNA with an amino acid, so that this tRNA can take that amino acid and attach it to a growing polypeptide. Unless, of course, this amino acid is methionine, and this is the start codon, and then it's not attaching this amino acid to a growing polypeptide. It's starting the polypeptide, right? We'll get to that in the very next question. All right, so here's a ribosome, and within this ribosome, you've basically got 3 spots for tRNA. You have the ejection site, you have the polypeptide site, and you have the amino acid site, or peptidyl site and aminoacyl site. But you could just polypeptide site and amino acid site. And so this is where your new tRNAs come in, and then your polypeptide gets transferred over onto that tRNA, and this whole complex moves. And once the tRNA ends here in the exit or the ejection site, it's ejected from the ribosome. Any questions about this, about the different RNAs? Okay, we're going to take another lecture break. And so what I'd like for you to do is I would like for you in your groups, take a couple of minutes and actually write out the sub-steps of the 3 steps of translation. Try to do it without using the slides. So there are 3 steps of translation. They are initiation, elongation, and termination. But there are sub-steps of those. Knowing what needs to happen at those stages, write out the sub-steps of translation. Okay, take a couple of minutes, and let's do this. Do it for all 3 steps: initiation, elongation, and termination. But I'm going to have your group give us the sub-steps of one of those 3. Okay, so make sure you have a good response for all of them. You guys lost another member. Wow, just group 2 is falling apart. Group 3 isn't doing much better, though. But you guys have a more systemic issue, I think, where you have people that just don't come. Ever. Not that come and then, leave a little bit. All right, take a couple of minutes, starting now.
What?
Small subunits.
Initiation.
For initiation.
We're talking about translation, right?
Yes, translation.
Small and large subunits. There's just direct.
Oh yeah, like stop codon.
Okay,
And
wait,
then
so,
they have to find the start.
right,
That's
so
in the
that,
termination. Yeah, small and large subunits of the ribosome. Yeah, there's
former, the right.
Yeah.
Okay.
And then that is the process. Well, they have to find the start codon. That's where they run.
Next, elongation. Starts with after that.
Okay, and then they have to find the start codon.
Reads.
We'll find the start with the ribosome. Ribosome has to bind with the start codon. Okay, well something has to get charged, right? The tRNA has to get charged with an amino acid. Yeah.
So it folds before that little loop.
Okay.
And then it
tRNA has to get charged with an amino acid.
But that's like separate from actual translation, because the tRNAs are just what's coming and bringing elongation. They just keep coming and bringing elongations. Before that whole process, they've already been charged.
You got about another minute. Make sure you can characterize the substeps.
Elongation.
Elongation and the tRNAs come into the
For elongation, you have to actually bind amino acids.
which are slowly added on according to the codon.
And they're getting recharged.
And then the tRNAs just get recharged into that.
tRNAs get recharged. Okay, what about termination?
They come to a stop codon.
They have to find the stop codon, so
Yeah. Once they reach the stop codon, the subunit drops off, and the amino acid changes.
whoa, subunit drops. And amino acid changes. Okay. So for initiation, small and large subunits gotta find. You gotta form a ribosome. Then you gotta find where the start codon.
Yes.
Then for elongation, you need the tRNA to come in there with the amino acids, and those are gonna get continually recharged. The anticodon combined with the codon. For termination, they need to find the stop codon. The subunits break apart, so the ribosome breaks apart, and the amino acid changes.
All right. We're going to start with group 6, and we're going to work our way counterclockwise. Okay, group 6.
I'm ready. We're ready.
What are the substeps of elongation?
Okay, so for elongation,
Yes.
you've already started, and your tRNA comes in with your amino acid.
Okay.
The anticodon binds with the codon.
Yes.
And after that happens, that tRNA needs to go back and get recharged with a new amino acid.
It sure does. What happens? Where does that tRNA enter the ribosome?
It enters in the thing that it enters in that was on that little picture.
Yes, the A site.
The A site.
Yep. It enters in the A site, and then the ribosome moves, and as it moves, it transfers the polypeptide that's on the tRNA in the P site over to the tRNA in the A site that now moves into the P site, basically at the same time. And then the next tRNA has to come, and yes, that tRNA needs to go and to be recharged. Love it. Group 5. Let's go with initiation.
Well, we said the TFs bind to the Tata box.
Well, so that's in transcription. So you've got initiation of translation. You still have TFs, but now you have translation factors, and they're binding as well, not to the Tata box. The Tata box is for transcription, but they're binding to the mRNA at very specific sites. All right, group 4. Let's go termination.
They hit the A tail.
Not usually. They usually won't get all the way to the poly A tail, and instead they get to a stop codon.
Stop codon.
Yep. They get to a stop codon, and there's something very interesting about stop codons. Group 3, let's go with termination. What's so interesting about those stop codons?
Well, they're like UA, UGA, UAG,
Yes?
and do they have an amino acid?
They don't have a tRNA that recognizes them, therefore they don't code for an amino acid.
Disengages the whole operation.
Yeah, so instead, since there's no tRNA with an anticodon for those codons, instead what's called a release factor binds, and the whole operation disassembles. The whole operation disassembles. So those stop codons do not code for an amino acid, which is an indication to you that there's no tRNA that binds there, and the whole thing becomes unstable. All right, group 2, the remaining 2 members of group 2. Initiation.
We said the ribosomes move down the mRNA and begin at the start codon.
Yeah, so they're scanning, right? And so overall in initiation, you've got your small subunit binds to the mRNA and scans it. So the small subunit binds, the first tRNA that has methionine on it binds in the P site, and they scan, looking for that start codon, AUG. And once they find that tRNA has like a GTP attached to it and provides the energy necessary to get the large subunit to attach, and now elongation starts. All right, group 1, do you have some thoughts to add on elongation?
It's like the reading and
Huh?
writing.
Yeah, and so you've got constantly just codon after codon translating from a word with 3 letters to a word with a single letter. And you have to keep in mind that there are some amino acids that correlate with more than one codon. And so there are some amino acids that occur at really high frequency because there are multiple codons that lead to them. All right, so the translation again occurs in 3 steps. Initiation, and this is aided by initiation factors, sometimes called translation factors, TFs. And so the ribosome moves down the mRNA until it finds the start codon, and that's just the small subunit is moving down the mRNA along with tRNA looking for that start codon, and that start codon is translated into methionine, and then that gets the whole complex to assemble with the large subunit, another tRNA coming, and everything transitions over to elongation, aided by other translation factors or elongation factors. And this, again, the ribosome is moving down the mRNA 1 codon at a time, every codon pairing with a specific tRNA. Every codon's pairing with a specific tRNA. And then that new tRNA basically transfers its amino acid to this polypeptide chain. But really what happens is that polypeptide chain gets transferred to that new tRNA with its amino acid. And so you keep growing your polypeptide longer and longer, one amino acid at a time, as you translate your word with 3 letters, your codon, into a word with a single letter, a particular amino acid. Okay? And then that tRNA, once it gets to the E site, so it comes in at the A site, the ribosome moves, now it's in the P site with the whole polypeptide attached to it. And then when the ribosome moves again, it transfers its polypeptide to the next tRNA, and now it's in the E site and gets ejected, and then goes and gets recharged. Okay, and they can do that thing all over again. And then finally, termination. The complex disassembles once you read a stop codon, and that's because there are no tRNAs for these stop codons. UAG, UAA, and UGA, there are no stop codons, there are no tRNAs that recognize those stop codons. So instead, again, a release factor binds there and creates an enormous amount of instability. Because now when the ribosome moves and it wants to transfer the polypeptide to the new tRNA, there's no tRNA there. There's no amino acid there to transfer this polypeptide to, so it frees up the polypeptide and the whole complex falls apart. Yeah, Chris?
So during elongation, is the peptide chain constantly changing between the A site and the P site?
Yes. Yeah, going from P to A, but at the same time that the mTRNA moves into the P site. So it's happening so quickly that if you were to actually watch the process, the polypeptide never leaves the P site.
Right, okay.
But yeah, it's transferring over to the A site at the same time that tRNA in the A site is moving into the P site. So it's happening basically at the same time. But it separates it out into individual steps for simplicity, but in reality, it's all happening simultaneously. It's all happening simultaneously. Now, initiation is a little bit different. Those steps really are happening in steps as it's scanning. But it scans really fast. I mean, it's moving, through thousands of codons a second. Yeah, Joey.
How long does it take
To make an
translate-
entire polypeptide?
Yeah.
Not long. I mean, a matter of a fraction of a second to maybe a couple of seconds, depending on how long the polypeptide is.
But is it still moving a lot slower than like DNA replication as far as like per?
Not really. No. Yeah. Not really. I mean, it's moving slower than replication in prokaryotic cells with a low level of fidelity, but it doesn't move really slower than copying the DNA in a eukaryotic genome. And you're like, well, then how does the DNA that's 4 billion base pairs long copy in a reasonable amount of time? And that's because it starts in literally tens of millions of places. So it doesn't like start at the beginning and run all the way through all 4 billion bases.
Yeah.
It starts at several million different places and runs until it's done. Yeah. Yeah, Charles.
tRNA, when it's scanning codons, just randomly pick amino acid and see if it fits, or how does it know?
No, yeah, it's an excellent question. Yeah, I don't know if we exactly have the answer to what gets the exact tRNA to be there and how this process can happen so quickly. Because there's only one. So the initial one, this is the beginning of initiation. Your small ribosomal subunit, along with this tRNA with methionine, starts scanning your mRNA, and they're looking for the start codon. So that's just going to go until all 3 of these bases can form its hydrogen bonds. But then this now, the GTP attached on here will be cleaved and will provide the energy necessary to attach the large ribosomal subunit. And now you have an A site that new tRNAs can come into. And then the question is, well, what keeps the wrong tRNA from coming in here? And the answer is probably nothing, but then it can't form hydrogen bonds, and so it doesn't stay. It's unstable. And so the only one that's going to stay is the one that's stable, and yet this is still happening at, several hundred, if not 1,000 a second, to where you're doing these translations codon by codon. And so, yeah, the answer is, I don't know if we know exactly what's going on here, other than that you'd probably get the wrong tRNA and it just wouldn't stick. It wouldn't form those hydrogen bonds. And I mentioned this before, if the bases don't hydrogen bond, they repel each other, right? So it's not like there's a neutral position, right? It's like we're good or we're okay or we're bad. It's you're good or you're bad. It's either it sticks and you form those hydrogen bonds, or it doesn't stick and you don't form those hydrogen bonds. And then so now initiation is done and elongation takes over. And elongation, you're just going one codon at a time. A tRNA comes into this A site. At the same time that it moves over, the polypeptide is transferred over to that new tRNA. And the old polypeptide is ejected. But this is all basically happening simultaneously. As the ribosome is moving over, the polypeptide moves over, and the other tRNA is ejected. All basically happening at the same time. Is it possible that they are happening in steps, step 1, 2, and 3? Sure, but it's happening so quickly there's no way you could see it as individual steps. What you would see when you watch this process is the polypeptide never leaving that P site, but tRNA is coming in and leaving. That's what you would see if you were to watch this. And so translation termination, against now this one does have distinct steps. Ribosome reaches a stop codon. That should say UAG, UAA, or UGA. I don't know why the G's disappeared from here. And so ribosome reaches a stop codon. There's no tRNA for that. So a release factor binds instead. And then when this ribosome goes to move, and at the same time that it would move, it would transfer this polypeptide. There's no amino acid there to transfer the polypeptide to. So the polypeptide gets released, and the whole process falls apart. But notice, still requires energy to split these pieces all apart. Every step in this process, every step in gene expression is energetically costly. Okay, every step in the process. Meaning, if you really wanted to control and not make a particular protein, you would want that to start with you never transcribed that gene in the first place. Right? So it gives you the idea that there's probably something going on at the chromosomal level to ensure that transcription doesn't happen unless you really, really want it to. Keep that in mind, store it up. We'll deal with that in a little bit. Yeah, Charles.
So in these illustrations, it looks like these things are suspended on nothing. And
The ribosomes?
yeah, well, the whole operation.
Yeah, we'll talk about that in the very next slide. Sometimes they are suspended on nothing. They're free in the cytosol, but a lot of times they're physically attached to another structure.
Including the tRNA.
I mean, the tRNA is attached to the ribosome and the mRNA. I mean, it's not just suspended freely, but the ribosome oftentimes is. That machine is suspended freely.
If it's suspended freely, what keeps it from bumping into all the cytoskeleton?
Chemical influences and chemical interactions with cytoskeletal elements. Yeah, I mean, nothing's completely free, right? It's in a chemical environment, rich in cytoskeletal elements. So nothing's completely free. But yeah. All right. So at the end of translation, what do you have? At the end of translation is done. What do we have? What are all the pieces that we have? Group 1. Polypeptide chain. That is one thing we have at the end of translation. Group 2. Something we have at the end of translation in addition to a polypeptide. Sure, well, possibly. It's not protein yet. It's a polypeptide. It's not quite protein yet, right? Because all you have is the primary sequence right now. Remember when we talked about proteins, we need a secondary and tertiary sequence at the very minimum, and sometimes that quaternary sequence. But you've got free tRNAs, right? The whole complex falls apart. So at the end of translation, you've got free tRNAs that can be recharged and can go again. Group 3, free ribosomes, right? You've got free small subunits and you've got free large subunits. Group 4, mRNA. You've got a free mRNA that can be translated by another ribosome. And we're actually out of components. I'm sorry, groups 5 and 6. It happens, right? Sometimes there's just not enough to go around. I'm sorry. These groups ate too much. All right, so what is left to make proteins? Because at the end of translation, we have a polypeptide. We have a polypeptide with a primary sequence, but it still needs to develop a secondary and a tertiary sequence at the very minimum in order to be a protein. All right, just a little nugget of truth. Many polypeptides can actually be made at the same time with the same mRNA. So what you'd find is a single mRNA may have 100 different ribosomes on it that are at different places in the process of translation. It's like if the cell went through the effort of making that mRNA, that was expensive to make. It had to write it, then it had to process it, shuttle it out of the nucleus. That was expensive. It's going to get its money's worth. It's going to make a ton of protein, if that's what it needs, if it needs that protein. Now, many, if not most, proteins actually need other proteins called chaperones or chaperonins to get the proper three-dimensional shape. The secondary structure, those alpha helices and beta sheets, some of those secondary structures, those will happen on their own because of how the amino acids interact with adjacent amino acids. But the overall three-dimensional shape of the protein usually has to be accomplished by other proteins folding that protein. And now you do have a chicken and egg situation, right? We eliminated that with the ribosomal RNA catalyzing, translating proteins. But we have a chicken and egg situation here with that proteins only get their proper three-dimensional shape by having other proteins help them in that process. And I love that we call these proteins chaperones, right? Ensuring that they behave properly. And now you do have a chicken and egg situation, right? What came first, the chaperone or the protein? And you're like, well, that doesn't even make sense. Chaperone is a protein. What folded that? Especially because when you look at chaperones, they have to be folded by other chaperones. And now you've got just a really interesting issue. So proteins that are going to actually work in the cytosol. So there are enzymes that will be in the cytosol for breaking down fuel molecules. You have proteins that function on the cytoskeleton, motor proteins. Remember we talked about those when we talked about the endomembrane system. It was like 3 weeks ago, and 3 weeks by this context is just an enormous amount of time, right? But yeah, any proteins that are assembled, that function in the cytosol or on the cytoskeleton, they are assembled on free ribosomes that in essence, functionally are floating in the cytosol that are separate. But proteins that leave the cell or function anywhere in the endomembrane system, these are translated on ribosomes physically attached to the rough endoplasmic reticulum. And that's what makes it rough, is that it's covered with ribosomes. And the reason it's covered with ribosomes is because those ribosomes are translating protein and at the same time shoving those proteins into the endoplasmic reticulum. This is good stuff. And I'd love to continue to paint this picture for you, but I'm out of time. So you have a wonderful conclusion to your Wednesday. We'll actually have to take a pause on this whole discussion of chapter 15 until Monday. And probably until like noon on Monday, because we have to talk about research abstract. Goodbye, everyone. Thank you for sharing your day with me.